Okay, guys, so today we are going to start learning about the topic which is mitosis. Now, mitosis, I gave you guys a little bit idea yesterday, is a type of cell division in which the number of chromosomes they remain the same. Now, we are human beings, right? And we have how many number of chromosomes in our nucleus? 46, right? So when the mitosis will take place, all the daughter cells, they will have how many chromosomes? 46. Does anyone has any idea how many chromosomes are present in chimpanzee?
52, sir.
48, right? Chimpanzee has 48 chromosomes. Now, you know that scientists, they are always experimenting on different things. And they are trying to make different kind of hybrid animals, right? And also they are trying to make new species by interbreeding different kind of animals, right? So, you see, like mule is a hybrid. We will talk about that when we will be learning about genetics in A2, inshallah, right? A hybrid is an animal which is not a species. Who will tell me the definition of species?
Group of organisms that can breed or produce fertile.
This is the answer, right? A group of organisms that can interbreed to produce fertile offsprings, right? Now, a hybrid of, you see, this mule is a hybrid in which it's a hybrid between a horse and a donkey, right? Now, this mule cannot interbreed. It does not have sex organs for that purpose, right? So we cannot call it a species, we call it as a hybrid. Do you think that scientists have ever tried to make the hybrid of the humans? Yes, definitely. But you see, these are classified things, right? Which does not appear normally into the journals. But in the early 1900s, some Russian scientists, they did try, but they did not become successful. Now, they did try to make it between a human and a chimpanzee. What do you think? Now, I am telling you that the chimpanzee, it has 48 chromosomes. Just think at these figures and tell me which thing is not making it possible to make a hybrid between a human and a chimpanzee.
So mitosis.
The number of chromosomes. First thing, how? When a gamete will be formed from the human being, it will have how many chromosomes? 23, right? And how many will be over here? 24. So when a zygote will be formed, it will have an unequal number of chromosomes, right? It will have 47 number of chromosomes, unequal, right? So that is not feasible for cell division. Understand? For cell division, for the mitosis to take place, there should be an even number of the chromosomes, right? We are going to learn about this today, inshallah. Now, first thing which we are going to talk about, that is cell cycle. Now, if you guys remember, I have taught you, whenever a cell reaches to its maximum size, then it has to divide, otherwise it will die, right? Now, there are different procedures, there are different methods, there are different steps through which a cell has to pass for it to divide. Now, it is represented in the form of a cycle. Now, let's suppose that this over here is mentioned as M. M is representing what? Mitosis, right? That the cell division has taken place, and with mitosis, they have mentioned another phase which is called as cytokinesis. The division of cell, complete cell, is called as cytokinesis. We are going to learn about it in detail. Now, once a cell divides by cytokinesis, Then a period of growth starts, which is called as either G1. In some books it is mentioned as G1, and in some books it is mentioned as G0. We do not call it G0. What do we call it as? G0. Right? In some books it is mentioned as G1, in other it is mentioned as G0. So this is the initial growth before the cell is going to divide. Growth means that the cell will elongate, increase in size. It will increase its number of organelles. So in the G1 phase, what is happening? First of all, increase in size of cell. Then increase in number of organelles. Right? So all these things that develop, like you see, a smaller cell requires less number of mitochondria. But when a cell will become larger in size, it requires more energy, so it will require more number of mitochondria. Smaller cell will require less number of endoplasmic reticulum. The larger cell will require more, right? So these organelles, they will increase in number. Larger cell will require more ribosomes for the protein synthesis, while the smaller cell will require less ribosomes. Now, once the G1 phase will complete, after that, the S phase starts. The question is, sir, what is S phase? Now here the letter S, it represents synthesis phase. Now what is the meaning of synthesis? To make, to produce, right? In other words, production. So what is produced in the S phase? In the S phase, replication of the DNA takes place. Now, if you guys remember yesterday's lecture, I told you that this is what? A parent cell. It divides to form two daughter cells, and both of them have equal number of chromosomes. Same number of chromosomes as the parent cell. So what did it tell you? That just before cell division, over here, replication takes place. And I told you guys that this replication word is derived from the word replica. And the word replica simply means copy. Right? So what happens over here? The replication of the DNA. Copying of the DNA. So this is the S phase. When the number of chromosomes, they will become what? Double than double. Right? They will become 92. This is the phase just before cell division. This is the phase just before cell division. Then there will be G2 phase. If we consider this one as G1 phase, this will be called as G2 phase. If we consider it as G0 phase, then what it will be called? G1 phase. It will be called G1 phase. So either we can call it G2 or G1 depending on what notation we are using, right? So what is the G2 phase? Now this is the phase where, let me mention over here, where proofreading of the DNA takes place. Now let's suppose I give you a page, exactly note it down. When you will note it down exactly, let's suppose I say that you see, note this lecture down. You are noting it down. Can you make some mistakes? Right? Yes, there can be some errors. Same thing, when replication is happening, there can be some errors in the replication. There can be some addition of nucleotides, or there is some deletion of nucleotides. That's not good. That's not good, right? Because that will change the genes. So during the G2 phase or G1 phase according to G0, what happens? Proofreading of the replicated DNA takes place. It's checked. Is there any problem? Right? If there is a problem, it is fixed. Right? So there is fixing of errors. So errors are fixed. Remember, this, you see this proofreading and replication of replication, and you see fixing the errors, it is part of replication process. Inshallah, we'll learn that in the next chapter. Right? So this takes place and cell further grows. And that will be the time when cell will reach to its maximum size, right? And after that, the M phase will start, the mitotic phase will start, the mitosis phase will start, right? Now, question is, does or do every cell divide? Answer is no. All the cells don't divide. Let's suppose, you see, when this cell, it divided, this parent cell divided to form these two parent cells. Let's suppose that this cell needs to divide, this cell do not need to divide. There is a fate of each cell, right? So the decision about dividing or not dividing is taken during the G1 phase. There are certain chemical changes that take place during the G1 phase that determine whether a cell is going to divide or not divide. Understand? Because after G1 phase, the S phase will start. If the cell needs to divide, then it will go into the S phase. If the cell do not need to divide, is there any need of S phase? No. Understand? So let me tell you over here that in the G1 phase, that cell division starting, what do we say, that this is cell division starting materials are produced, or in other words, chemical changes that determine the fate of the cell, right? Whether the cell is going to divide or not. If the cell needs to divide, then it will go into the S phase. If the cell do not need to divide, then it will not go into S phase. Now guys, this M phase is further divided into 4 stages. M phase is further divided into 4 stages. Now remember that. This mitosis, it's a continuous process. It will not happen that you see, first this stage will complete, then this will, then this, then this. It's a continuous process. But for our understanding, we have divided it into 4 stages. First stage is prophase. First stage is called prophase. Second stage is called as metaphase. Third stage is called as anaphase. And the last stage is called as telophase. And then cytokinesis takes place. Right, guys? Now, before we start learning about these phases or these stages, let me remind you the structure of the cell, only the parts which are related to the mitosis. Only the part which are related to the mitosis. We have learned all these things in chapter 1. So guys, let me draw a small cell. What is this? This is cell membrane. Then, in the middle over here, we have the nucleus. And in the nucleus we have what? What is this called, boys? Who will tell me? Very good. This is chromatin. Remember, yesterday I told you, if I'm drawing like this, I'm representing chromatin. Now, what is the difference between chromatin and chromosomes? Question can come. So, what is this? Yesterday I told you the difference between chromosomes and chromatin. Yes.
Protein is made from DNA and histones.
Okay, and
Chromosomes are only made from DNA.
chromatin is loosely packed DNA with histone proteins, while chromosomes are tightly packed, supercoiled DNA and histone proteins. Understand? Difference is that this one is loosely packed. It's loose. But when it will become chromosome, it will be tightly packed and it will be supercoiled. Remember that? Right? So that is the difference. Now who will tell me what are these two structures that I drew over here? Nucleoli. Nucleolus is singular. This is nucleoli. Then another structure which I am going to draw over here. What is this structure? Yes. Centrosome.
The area or the structure?
Whole structure with the surrounding dense cytoplasm is called centrosome. Understand? But only the structure is called centriole. Remember that, right? So this over, and even you see right now I drew this dense cytoplasm around it. So what is this? This is centrosome. Now, for mitosis, only these structures which we need to discuss. That's why I only drew these ones, right? There is cell membrane, and right now I am talking about animal mitosis, animal cell mitosis, right? At the end, we'll talk about the plant cell mitosis as well. All these stages in the plant cells are also exactly the same, but there are few differences which we'll only discuss at the end, inshallah. Now, so what is it? Cell membrane, nucleus, and what is the name of the covering of the nucleus?
Nuclear envelope.
Very good. Nuclear envelope, or we can call it nuclear membrane as well. Right, guys? Now, this is before cell division, right? So the nucleus has its nucleolus or nucleoli as well, and chromosomes are in the form of chromatin. Remember, I told you, when they are in the form of threads, these chromatin form, it is very difficult to separate them from each other. But it will become easy when they become what? Compact. Remember that? So what is the first stage should happen? That these chromatin, it should change into chromosome form. It should change into chromosome form. Now here I have another question. Who will tell me what are nucleoli made up of? Someone's mic is on. Can you please keep your mic mute? Yes.
So you said satellite DNA exactly.
Satellite DNA, right? These nucleoli, they are made from satellite DNA, but what is their job? rRNA, right? They have the genes for ribosomal RNA, right? So remember that the nucleoli, they are made from satellite DNA. And they have genes for ribosomal RNA. Remember that I told you guys, whenever we write ribosomal RNA, we represent it with small r for ribosome and capital R, capital M, capital A, right? So this is ribosomal RNA, right? So they make ribosomal RNA. So as this is the satellite DNA, and satellite DNA is part of chromosomes, right? So it means that when these nucleoli, they are visible, these satellite DNA, they are compact at one place, right? They need to go back where? To their original chromosomes. To their original chromosomes. So when this chromatin compaction will take place, these nucleoli, they will go to their original chromosomes. So will the nucleoli be visible at that time? Answer is no. So in the prophase, let me write over here. I have made the notes as well. I will share the notes with you guys, right? Each step, it will become very easy for you to memorize. So in the prophase, first step, what happens? This chromatin condenses and it changes into chromosome forms, right? So we mentioned here what happens. Chromatin condense and change into chromosomes. As I just explained you, that these nucleoli, they were part of DNA, so they will also disappear during the prophase. So what is the second step? That the nucleoli, they disappear. Understand? Another thing, that this, you see, when there will be chromosomes, these chromosomes needs to go to the opposite poles, right? For that purpose, they needs to be pulled. And if you remember the first chapter, we have learned what is the job of the centrosomes?
Spindle fiber.
Formation of spindle fibers. Formation of spindle fibers. Normally, in one animal cell, there is one centrosome. But for cell division, there will be requirement of what? Two. So, in the prophase, what happens? That centrosome replicate and make another and makes a pair, right? So what happens? That centrosome replicate and make a copy of centrosome. Now these centrosomes, they should be on opposite ends of the cell, right? So what will happen? In the fourth step, centrosome moves to opposite poles, right? Opposite poles means opposite ends of the cell. Next step, what will happen? When these chromosomes, I am going to draw the diagram as well to show you, inshallah, don't worry. I will represent everything with the diagram as well. The chromosomes, when they will be pulled on the opposite sides, will there be any nuclear membrane there? No. Right? So another step, what happens? Nuclear membrane disappear. So two things, they have disappeared. Nucleolus or nucleoli and nuclear membrane. one thing has replicated, centrosome, and it has gone to the opposite ends, right? Now let me show with the help of the diagram. Now again we have here the nucleus. In the nucleus, I am showing you guys these 4 chromosomes. Now they are in compact form. Before they were in which form? Chromatin form. So what has happened? Chromatin condensed to form these chromosomes, right? So this diagram is showing that chromatin is changed into chromosomes. Next thing, what did I tell you guys? That what happens that the centrosome, it replicates, right? At these centrosome, they will move where? To the opposite poles. This one will come over here, this one will come over here. Understand? And what did we learn? That the nuclear envelope starts to disappear, and it completely disappears at the end of the prophase. Now, how can we show the disappearance of this nuclear envelope? I can make another diagram for that purpose. First, you see, I am drawing the nucleus exactly the same way. And we have these chromosomes over here. And I can also show that these centrosomes, they have reached where? To the opposite poles in the next diagram. Right? Nuclei, they have disappeared, and nuclear membrane is what? Disappearing. Got it, guys? So when the nuclear membrane will be completely disappeared, at that time prophase will complete. Right? So let me mention over here that when nuclear envelope completely disappear, prophase completes. Understood, everyone? Now, I can show that in another diagram as well, very quickly, the completion of the prophase. Now, there is no more nuclear envelope. There are these chromosomes over here. No more nuclei over there, and there are what? These centrosomes are present. Got it, guys? Now, next phase. Which one? Metaphase is going to start. Next phase, metaphase is going to start. First of all, let me write down what happens during the metaphase. First of all, centrosomes, they have reached to their opposite ends. So what will the centrosomes do? They will start making spindle fibers. Now remember that spindle fibers, they are made up of microtubules. Now when I taught you about the cytoskeleton in the chapter number 1, I taught you that they are made up of, you see, there are 3 types of tubules: microtubules, intermediate filaments, and microfilaments. Remember that? So these here these spindle fibers, they are made up of microtubules. Understand? So let me mention over here, first of all, what happens? That centrosomes start making spindle fibers. They have started making the spindle fibers. Second thing, the chromosomes, they arranged on equator. Do you know what is equator? Center. Right? So next step, what happens? That the chromosomes, they arrange at equator. Equator means right in the middle. So boys, look here. This is the cell, right? And the chromosomes, they will arrange like this. Got it? Now, and on these sides, what do we have? We have centrosomes. Now these centrosomes, they are making what? Spindle fibers. So the blue lines, they are representing what? Spindle fibers. Now these spindle fibers are of 2 types. If you see this spindle fiber, it is starting from here and ending at the other centrosome, right? So this kind of spindle fiber will be called as a continuous spindle fiber. While this spindle fiber is starting from this centrosome and ending and attaching to the centromere of this chromosome, right? So is it attached with the other centrosome? No. It is stopping in the middle and attaching to the centromere of the chromosome. So this kind of spindle fiber is called discontinuous spindle fiber, right? So let me mention over here. So this one is continuous spindle fiber, and this one over here is discontinuous spindle fiber. That was. Now, all these spindle fibers, they have attached to the centromeres of the chromosomes. So we can mention this point as well over here, that spindle fibers attach to centromeres of chromosomes. Just one point came into my mind. Yesterday we did not have enough time, so I could not finish that. Remind me in the next class, I have to teach you about sister chromatids and non-sister chromatids, okay? And homologous chromosomes as well, inshallah, right? So guys, spindle fibers, they attach to centromeres of the chromosomes. Now what should be the next step? Pulling them apart. Now remember the yesterday's lecture. I told you that this is not one chromosome, this is actually 2, right? This is after replication, after making the copy, right? So then next phase starts. And what is the next phase? Anaphase. In the anaphase, what will happen? That these, you see, spindle fibers, they will start contracting. These are microtubules, right? They can contract. They will start contracting and they will start pulling the chromosomes to their ends, towards the opposite poles, right? So what do we mention over here? that microtubules of spindle fibers, they start contracting and start pulling chromosomes towards opposite poles. Now we can represent this in the form of this diagram. Now what has happened? The chromosomes which were duplicated, they have become separate from each other. They have become separate from each other and they are pulled in opposite directions. This one is being pulled in this way and this one is being pulled in this direction. Got it? Now, this is the anaphase. Then the last phase will start, which is the telophase. Now, in the telophase, what happens? That these chromosomes, they will reach to the opposite poles. They will reach to opposite poles. Now, once they will reach to the opposite poles, now the chromosomes, they are totally separated, right? In equal numbers. Now, what will happen? Reappearance of the nuclear membrane will start. Reappearance of nuclear membrane will start. So we can first of all write down over here, or first of all let me draw the diagram, then I will write on the side because we don't have enough space. That now we have this. And this. So these chromosomes, they have reached to the opposite poles. Now the nuclear membrane starts to reappear. I am showing it with the help of this dotted line. I am showing it with the help of dotted line, and eventually this, I will draw a small diagram over here of the end of the telophase, that two complete nuclei will be formed. What do you see? Two complete nuclei formed, right? And they have those 4 chromosomes. Right, guys? So we started with 4 chromosomes and we have ended with what? 4 chromosomes. Now in 2 nuclei. Right? So we can mention over here with the telophase. So what happens? That first of all, that chromosomes, they reach to opposite poles, right? And nuclear envelope reappear. Yes.
So we started with 8 chromosomes.
This is not 8. These are 4 chromosomes but duplicates. Right? Remember this phase, S phase, right? These are duplicates, right? But you see from the G1, this cell which was at the G1, it is actually divided, right? So this one had 4 chromosomes, let's suppose. Over here it became 8 just because of replication, right? And then when it divided again, 2 daughter cells have what? 4 each. Understand? So this is just after the S phase and G2 phase, right? So when this whole cycle completed, at the end what we have again? 4 chromosomes in each nucleus, right? So here it is the end of the telophase. And then what will start? Cytokinesis. How much time do we have? 15 minutes, right? So we can do that. So I am going to rub from this side, okay?
Sir, I have some questions.
Yes, sure.
Sir, how is this all observed?
Observed?
Yes.
Yes.
Spindles are smaller than to be seen from
Yes.
a light microscope. So
No. Very interesting question. Look here. I explained this point yesterday. When chromosomes are in the form of chromatin, they are not visible, right? But when they become condensed, I told you guys this categorically, if you remember yesterday's lecture. I said chromosomes are only visible just before cell division and during cell division, right? Now, interesting fact, where did I write the word chromosome? Where? Did I write? Let me write over here. Chromosome. How did the scientists discover it? Because, and you see, how did they give it name? Chromo and som. These are 2 words. Chromo means color. And som is the word derived from a biological word soma. Now in biology, wherever you see the word soma, it means body. Colored bodies. So during the cell division and just before cell division, with special kind of dyes, the scientists, they were able to see the chromosomes because it is the stage when they are in compact form. It is the stage when they are in compact form, right? So you see, they are, these all stages, they are visible with light microscope. These all stages, they are visible with light microscope. We can observe them because chromosomes become visible during and just before cell division. Got it?
yes, sir, another question is
Yes.
mitotic spindle the same thing as spindle?
So which mitotic spindle? Yes, exactly the same thing. You call them now. We also call them as MPOC. Mitotic or mitotic organizing center. Right? These are, you see, this is one MTOC, one mitotic organizing center. This is the place from where spindle fibers, they are arranged, they are organized, right? So mitotic spindles, or simply call them as spindle fibers, is same thing. Next thing, why are they called as spindle fibers? Now spindle, you see, is like, you see, it's making of an old method of making of threads. And when they are making threads in old method, they used to take a, ball of cotton in their hand, and they had, maybe you guys are not that much old. You see, when I was old, when I was young, I was a little kid in 1920s. So my grandparents and my parents, they used to tell me the story that there is an auntie living on the moon, right? And she has a spindle, and with the help of spindle, she is making the threads for making the clothes. So I will show you guys the picture of the spindle, inshallah, on Google. So it's a device with which they rotate it, and they are holding this up, like a ball of cotton, and cotton is pulled by itself towards the spindle machine, and it forms a thread. So under the microscope, they appear like that. Like you see that in olden times when the spindles, they were formed for making the thread. So that's why they have given it the name spindle fibers. Understand? So you say mitotic spindle or spindle fibers, this is the same thing. Any other question? Okay. So guys, till now what we have done, that what has happened? Two nuclei are formed. But cell is still one. This cell will divide, then two cells will be formed, right? Now when this cell will divide from the middle over here, then what will happen? Not only the nucleus will be divided, cell organelles will also be divided. Cell organelles will also be divided. And that whole process, that whole phenomenon is called as cytokinesis. Now for that purpose, I am going to rub this area to show that. Today only we will talk about the cytokinesis of the animal cell. Inshallah, tomorrow we'll finish this chapter. We'll talk about the cytokinesis of the plant cell, and we'll talk about the cancer as well tomorrow, inshallah. And today I will share with you guys notes of this mitosis, in which you see there is a reference given of Henrietta locks. You guys need to check that today, okay? This is very, very interesting. It will be written Immortal Life of Henrietta Locks. A woman, she never died, but she is dead. How she is living? She is living all around the world right now. She was patient, she was an American, and she had cervical cancer. You know what is cervix, right? So they took the cancer cell of that woman and they started researching, and they took those cells without her consent at that time, and without knowing her family about that. And now you see all around the world, in Germany, in UK, in Korea, in all the well-developed countries, wherever there is research of cancer is going on, they have their cells, because cancer cells are keep on growing, right? So they have those cells in a continuous growth pattern in a certain artificial this environment. So come over here, boys. Now we have a cell with two nuclei. Right? Now, both of these have now equal number of chromosomes. Another very important point just came into my mind right now, which you need to know. Now, when the nuclear membrane will reappear, is there any need for the chromosomes to remain visible? No. So what will happen? The chromosomes, they will start changing into chromatin. At the end of the telophase, chromosomes will start changing back into chromatin. So they will start, even the chromosomes, they will start disappearing. Right? We will not be able to see at the end of the telophase. Then, what happens over here? Here, formation of spindles, special microtubules in the middle over here. Formation of microtubules in the middle over here. What does it do? Those microtubules, they contract from the middle, these microtubules, and what they will do? They will bring the cell membrane near to each other, like this. Right? So what is happening? That this microtubule, these microtubules which are present from one end to the other end, they will start contracting. When they will start contracting, what will be formed? A furrow will be formed. Right? This is called as a furrow. Right? Now, this furrow will keep on increasing, right? Remember that you see on the both ends there is nucleus, and this nucleus contains what? Equal number of chromosomes, whatever we have discussed till now. Eventually what will happen, that this you see, it will come very close to each other like this. It will keep on coming closer, right? Keep on coming closer, and eventually what will happen? Just like we have learned the topic of endocytosis, right? When 2 points they join together, eventually what happens? It splits up. So when they will both join together in the middle, it will split up. Now, what has happened? Cytokinesis has taken place. Cell is now divided into 2 cells, right? Now, there is division, not only division of nuclei, there is division of cell organelles as well. Now, what has happened? Now, the mitosis is complete. Now, again, I told you what will happen. Each cell will go into which phase? G1 phase. Now, it will grow. When it will reach to a maximum size, and I told you during this G1 phase, it will be decided whether this cell needs to go into next cell division or not. Let's suppose if this cell needs to, both of these cells need to go into next cell division. During that, certain chemicals will be produced, certain proteins, certain enzymes will be produced, right? And then it will go into the S phase, right? During that, replication will take place. Then G1 phase. During that, what will happen? This proofreading of the replicated DNA and fixing of the errors, and cell further will grow. And then again, this you see, mitosis will start. So what is happening? Cell is dividing again and again with the same pattern. So as it is dividing again and again with the same pattern, that's why it is referred as cell cycle. That's why it is referred as cell cycle. Understood, everyone? Any question was? Yes.
So where did the membrane, this nuclear envelope go when it disappears?
There are lysosomes, right? They, you see, make it disappear. They digest it. They break it down.
And then
Okay.
how it comes back?
Sorry?
Then how it comes back?
Again, why? You see, you know that there are Golgi apparatus, right? So Golgi apparatus makes the small vesicles, and those vesicles join together to make the cell, this nuclear envelope reappear, right? Small vesicles inside the cells, they will join together to make these nuclear membrane reappear. Got it?
Yes.
Yes.
I have a question. In mitosis, won't the mitochondrial number increase?
It increased over here. I told you that you see during this the number is increasing.
No, sir, before it happens, won't it exponentially increase because more energy is required for the spindles?
Definitely. See, that process takes place over here and here during the G1 phase and G2 phase. Alright? Any other question was? Done? Yes.
With this